Sounds great. Hi, everyone. Thank you to each and every one of you for joining us today to learn a little bit more about our data analysis and interpretation platform. We truly appreciate it. My name's Kim Waller, and I'm the VP of Sales here at Geneyx. And I'd also like to introduce my great colleague, Dr. Eli Sward. He's the Director, Field Application Scientist, and he'll be presenting today. So before we get started, sorry, Eli, before we get started, sorry. Yeah, before we get started, I just have a few, items to cover, and then I'll hand it over to Eli. Feel free to ask questions. There's a Q&A section down at the bottom. Just feel free to write your question in there. And if we have time at the end of the presentation, we'll definitely answer those. If not, we will definitely get you the answer and email it to you afterwards. We do have a poll down at the end. Definitely click on the poll and we'd love to get your answers. It just will help all of us. So if you have time, the poll would be great. I am going to record this. So we're recording this webinar and I'll send you guys, as soon as it's finished, I'll send this to you so that you have it as a copy. Also, don't forget, we do have free demo trials. So after listening to Eli present, if you have some challenging cases or you just want to learn a little bit more, write a few samples through us, feel free to reach out. I have our email addresses down here at the bottom. So please feel free to reach out at any time to ask additional questions afterwards. So I will hand it over to Eli.
Perfect. Thanks for that introduction. As Kim mentioned, my name is Eli Sward, and today what I'm going to be talking about is the Geneyx Analysis platform and really how this is a comprehensive workflow solution for your bioinformatic pipeline. Just to kind of give an intro into the company, so Geneyx is really composed of highly skilled individuals that have a passion for clinical genetics and improving patient care. If you were to look at our biographies, generally the average kind of experience in the clinical workspace ranges from 15 to 20 years. So we have a lot of experience that we are definitely implementing into our software, but we also have headquarters in Israel and the United States. So really what that means is we can offer support for the variety of different time zones that are available. But with the highly skilled individuals that are on this team, what we're really trying to do is kind of implement our vision for a clinical genetic software. And so what we're trying to do is really drive the future of the clinical genetic data analysis. And what we're ultimately doing is we're trying to automate the diagnostic process from sequencer to report using machine learning, as well as kind of an AI-based genetic analysis platform. But one of the things that we also are striving to do is to also create a resource for discovery. So in addition to being able to analyze your variants, we also want to support an environment where you can identify these novel mutations and ultimately lead into specific maybe publications. And a testament to our vision is really our customer base. So we have over 80 customers and we have close to 100 publications. Over 100,000 of cases have been analyzed with our software. And so the customer base is really, composed of top tier research institutions, governmental organizations, research, clinical settings, hospitals, diagnostic labs. Really, there's kind of a diverse field in terms of our customers. But one of the things that is underlying these is they all are using the Geneyx Analysis platform for their NGS data analysis. And so I wanted to just kind of take a couple minutes to talk about the software. So the Geneyx Analysis software can be broken down into a couple different categories. So this includes basically the ability to implement the primary all the way to tertiary analysis. So we can start with your raw data processing. We have flexible workflows. We also have seamless integration with bioinformatic pipelines, and we also have excellent reporting capabilities. And so just to kind of go into these in a little more detail, the raw data processing, really what that means is we have simple integration with variant calling pipelines. So you have the ability to utilize the Geneyx Analysis platform, starting with the FASTQ or VCF files. But one of the great utilities of the software is that we've really tried to optimize the variant annotation, especially for high throughput environments. So for targeted gene panels and whole exomes, it takes around 5 minutes to annotate these samples, but then for whole genomes, including the single nucleotide variants and CNVs, this can take 15 to 20 minutes. So it is a scalable solution. In terms of the flexible workflows, one of the things that is of primary importance in the clinical and diagnostic settings is obviously obtaining consistent results. And the way that you do that is by utilizing filter protocols. And so the filter protocols within the Geneyx Analysis platform are entirely customizable. And what they allow you to do is obtain those repeatable workflows. Furthermore, within the Geneyx Analysis software, it offers a flexible design layout. So if there's a particular feature that you want to have displayed when you open up the application, you have the ability to kind of create that modification. Furthermore, we support the ACMG and AM guidelines. So these are approved for CLIA-CAP genetic labs. The ACMG and AM guidelines can also be modified. So if there's a particular threshold based on a frequency or an annotation source, you have the ability to integrate that into your workflow. But more importantly, one of the things that you're able to do is store your interpretations for downstream case analyses. So if there is an interpretation or an evaluation made on a variant or gene level, and you see that in a future case, you'll be presented with your interpretation. And together what this does, it allows an easy automation of your NGS panel workflows, whether that's targeted gene panels, whole exomes, or whole genomes. In terms of the integration, so we have a flexible deployment. So in terms of installing the software, this can be installed on cloud. So we support Azure, AliCloud, Amazon, as well as some of the other ones that are out there. But it also can be installed on premise behind a firewall. So it is kind of a flexible deployment. In terms of the integration, we do have clinical labs that have integrated our software with their laboratory management systems or electronic health record systems. But we also support exporting of reports to external reporting engines. So we do have a couple customers that are direct to consumer. So if you wanted to integrate the Geneyx Analysis platform in terms of the reports, we do have the ability to kind of create a unique environment that allows you to obtain that. And lastly, security is of primary importance. So we are HIPAA and GDPR certified. Our data is backed by encryption, so you can ensure that your data is not being accessed by some third parties. And lastly is our reports. So a lot of different diagnostic and clinical labs will tend to use different, third-party reporting capabilities. And so, Geneyx Analysis is really a complete clinical solution. So we do offer reports. They come in a variety of different languages, and they're also, it has the ability to automatically incorporate the clinical and patient data. We do have a team that is dedicated specifically to performing customization on your reports. So if there's a particular lab design or brand design that you want to have implemented, we do have the capacity to do that. And lastly, as I mentioned before, these can be easily integrated with LIMS or electronic health record systems. And so with that kind of introduction, what I wanted to do is just change the gears a little bit and talk about some case studies that show the value of the Geneyx Analysis platform. And so one is by the Bambino Gesù, which is the largest pediatric research center in Europe. And Dr. Antonio Novelli's group adopted the Geneyx Analysis platform. And what they were able to do is they were able to drastically increase the number of cases that they analyze on a monthly basis. So maybe using their own bioinformatic pipeline, they were able to process around 12 samples per month. But when they adopted the Geneyx Analysis platform, that increased to around 1,000 per month. And so in turn, they also identified that they had a 65% diagnostic yield with a turnaround time less than 10 minutes per case rather than hours. And this is also has been a testament from our other customers, but ultimately this is kind of indicating that the Geneyx Analysis platform is a scalable solution, especially for those high throughput environments. I also wanted to highlight a pilot study that was performed with PacBio. So PacBio had 81 undiagnosed rare disease families from short read whole genome sequencing. And what they did was they implemented their long read hi-fi whole genome sequencing, which for those of you that are maybe unfamiliar with this type of sequencing, it sequences read lengths to around 50 kb, as opposed to short read, which is around 250 base pairs. And so this allows for better alignment, mapping to the reference genome, as well as a higher precision and accuracy in calling a variety of different types of variants. And so with this long read data, they wanted to test the different software solutions in terms of their analysis capabilities. And Geneyx was fortunate enough to be included in this pilot study, and we actually were able to achieve the baseline of the candidate variants, but we outperformed all of the other software solutions by identifying 15 additional cases for new and promising candidate variants. And so the conclusion that we basically extracted from this pilot study was that Geneyx is really a flexible NGS analysis solution that can resolve the challenging undiagnosed cases, but it is also valuable because it can work with a variety of different file formats. So this includes targeted gene panels, whole exomes, whole genomes, as well as short read and long read data. So this really kind of brings us to the differentiation. What we like to highlight with our software is we have the ability to reduce your turnaround time by 95%, as well as increase or improve your diagnostic yield to around 65%. And really, these are kind of generic blanket terms, but what I wanted to do is kind of provide a little more details in terms of how this is being accomplished. So in terms of reducing the turnaround time, really what we're talking about is improving efficiency. So as mentioned, we have easy integration with primary and secondary pipelines. But one of the things that is a great utility of the software is the ability to batch upload and perform audit trails. So for high throughput environments, you have the ability to upload multiple samples at a time. But we also have really nice management capabilities where if you wanted to get information in terms of when a case was imported, what genetic model or protocol was used, if a modification was made, or even when a report has been generated, we have really nice audit trails that give you and provide you that information. Another thing that is a nice feature about the Geneyx Analysis platform that improves efficiency is we stay up to date on our annotation sources through an automated process. So for the bioinformatic, maybe your internal bioinformatic pipelines, you understand how tedious it can be to stay up to date on these annotation sources. And so through an automated process, we alleviate that complexity and provide you with all of the relevant up-to-date annotation sources within the software. Furthermore, we automate the ACMG guidelines. So this was a publication that started in 2015, and they basically outlined 28 criteria. 18 of those are automated within the Geneyx Analysis platform. And then lastly, we have the highly effective gene phenotype prioritization tool. And this is actually what is allows us to improve the accuracy. So the phenotype-driven interpretation algorithm, what it does is it integrates biomedical knowledge bases containing comprehensive information on all human genes and diseases. These here are listed, or just a couple of the annotation sources that are implemented into this phenotype-driven interpretation algorithm. On the left-hand side, you can get just kind of a brief snapshot of what it's doing. Essentially, it highlights the key search terms. So it uses your matched phenotypes that you've entered. And then it highlights those search terms across the biomedical knowledge bases. And so in this example, you can see we have information being pulled in from OMIM, ORPANET, and UniProt, and it's highlighting those key search terms. And so for a clinical geneticist or an expert, we provide you with kind of a consolidated view of the information. So if you wanted to go in and deep dive a little further, we provide actionable hyperlinks where you can go in and read those publications. But the nice feature about this too is it outputs a score prioritized gene list according to that phenotype and symptom relationship. And so through integrating this type of algorithm, we've really been able to increase the phenotype-driven interpretation from 2 to 20%. So it's a pretty significant increase and allows us to increase your diagnostic yield. So changing gears, what I wanted to do is kind of talk about the project demonstration that I'm going to be focusing on. And we have two cases that we're going to be going through. So the first case is really a simple case diagnosis. This would be for the environments that are trying to process large sample volumes and you need a fast, scalable, robust solution, where you're also maybe focusing on clear-cut diagnosis, such as maybe a targeted gene panel or a whole exome. And so what I'm going to focus on with this case is a trio workflow. So we have mother, father, as well as a proband, and the proband is affected by tuberous sclerosis. And then the second case is going to be demonstrating challenging undiagnosed case. And so this may be for the clinical geneticist or the expert that is performing maybe whole genome sequencing routinely for undiagnosed cases. And ultimately what we're trying to advocate is, maybe you're not looking to replace your current pipeline, but interested in other solutions for difficult cases. And so Geneyx Analysis can definitely tailor to both of these environments. But ultimately for this undiagnosed case, we're going to be looking at the affected and unaffected siblings associated with Parkinson's. And so just to kind of talk a little bit about the first case that we're going to demonstrate, this is, again, tuberous sclerosis. And tuberous sclerosis is a rare autosomal dominant genetic disease. It causes benign tumors to grow in the brain and other vital organs. One of the key distinguishing characteristics about tuberous sclerosis is the skin abnormalities. So you can see the image on the right. We have these benign tumors that are growing around the mouth and the nose. It's also kind of a detrimental disease because it can impact the central nervous system, resulting in seizures, as well as intellectual development and autism. But for those of you that are familiar with this, it is a very straightforward case because it's generally only caused by mutations on two genes, which is the TCS1 and the TCS2. And so with that, I'm going to jump into the project demonstration. So as mentioned, the Geneyx Analysis platform can be installed on a cloud or on-premise. But ultimately, just to provide you with the general overview, we have the dashboard view here on the left-hand side. And in the dashboard view, it breaks it down into your analyses. So the VCF samples that have been imported into a particular protocol. And then you also have your VCF samples. One of the things that I like about this software is the ability to search for particular cases. So if I wanted to look at maybe all of my trio cases, it provides us with those relatively quickly. Another nice utility about this is on the left-hand side, you have a navigation pane. So in the analysis window, you have the ability to go into a little more detail in terms of looking at the analyses that have been imported, the VCF samples, the reports that have been generated, as well as the subjects. The data management is used for processing FASTQ files. So you have the ability to upload single samples as well as performing batch uploads. And lastly, you have the settings options, which allows you to kind of go in and modify protocols, gene panels, incorporate variant maps, etc. And this will be the focus of kind of future webinars. But for now, what I want to briefly start with is, let's say you wanted to start from scratch. Maybe you're new to the software. And the question is, how do you get your data imported into the software? So you can do that by clicking on the Start New Analysis. And this will bring you to the dialog of the available protocols. So as I mentioned during the presentation, it's of primary importance to obtain consistent results using repeatable workflows. Those workflows are defined ultimately through the protocol interface. And so we provide a couple protocols by default. This includes the germline, somatic, and we have some for health screening. And as mentioned, you do have the ability to create your own customized protocol, and you can then use that for all downstream analyses. I'm not going to go through the import process, but I just wanted to show you the protocols that were available for those users that are maybe new to the software. So coming back to the dashboard view, what I'm going to do is open up the tuberous sclerosis case, and all I have to do is click on the Analyze option here. And so what this is doing is it's going to load the variant dashboard. So the variant dashboard includes information that's being pulled in from the VCF file, as well as the different genetic models that have been applied. And so one of the great utilities about this is it has the ability to, it's very intuitive in terms of the information that is being displayed. And so once this loads here, so just kind of a general overview. In the upper right-hand corner, we have the VCF samples that have been imported into the software. So again, this is a trio workflow where we have the proband, the father, and the mother. With this, you can see the variants and the amino acid change for each samples. And we also will provide the identity by descent. So the identity by descent really allows you to validate if the mother and the father are true parents to the proband. And the way that we're doing this is by looking at the homozygous alternate in one of the parents and homozygous reference in the father, and then determining how many heterozygous variants are transferred to the proband. So you would expect for these two cases to have around 100%, and in this case, it is valid. So we can confirm that these are indeed the mother and the father, or it provides us some supporting evidence. In the clinical tab, it gives you a breakdown of the phenotypes that you've entered in terms as well as the specificity. So our phenotype is tuberous sclerosis. We have the applicable filters, the protocol that was implemented, as well as if maybe this case was assigned to a particular user. And then the subject will pull in relevant details in terms of the patient and clinical information. So beyond this, we have the variant dashboard, which is broken down into the different genetic models. So in this case, we have fast track, but we also have those that may be other inheritance patterns. The fast track is a great utility, especially for those simple case diagnosis, because it allows you to rapidly identify the candidate variants, because we will list those at the top. So just to give kind of a general introduction, here we have our variants listed on a per row basis. We have relevant annotation sources in our column view. Any column that's present, or any column that is present in your variant dashboard, can be filtered on, and the filter logic is displayed here on the left-hand side. But just to give kind of a general introduction in terms of the information that's displayed on the variant level, the location allows you to view the information in genome browse. If you have associated your BAM file with your VCF samples, you can look at the coverage and pileup information. You can also hyperlink out to the UCSC genome browser. We also will display really nice information on the gene level. In this case, we can see here that we have the TSC2, which, as I mentioned during the presentation, is one of the primary genes causing tuberous sclerosis. But we can see it's autosomal dominant. If we select this option, we can view relevant information that's being pulled in from OMIM as well as Decipher. And in this case, we can see that tuberous sclerosis 2, it's associated with an autosomal dominant disorder, has been known to be associated in OMIM with this particular gene. We can also see similar findings in Decipher. If we scroll down, we also display the summary of pathogenic and likely pathogenic SNVs in ClinVar for this particular gene. We can actually see that tuberous sclerosis 2 is associated with a variety of pathogenic and likely pathogenic variants in ClinVar, as well as 55 that are intronic, which is actually the same effect of the variant that we're looking at in this particular case. We will also display those pathogenic and likely pathogenic for CMVs in ClinVar as well. Beyond the gene level, we will pull in relevant information on the genomic and genetic data. So for example, one of the things that we will do is display a couple fields by default. But if you wanted to expand and view that information in more detail, you do have the ability to do so. As mentioned during the presentation, we also support the automation of the ACMG guidelines. So in this case, we know that we're looking at an autosomal dominant gene. So if we wanted to look at the dominant inheritance pattern, we can see it's pathogenic. Hovering over this will show you the applied criteria. In this case, it's PVS1, PS2, and PM2. But if we wanted to click on this event for more details, we can see the criteria that's applied, as well as the relevant variant information that is displayed on the right-hand side. Now, one of the great features about this too is if you wanted to disagree with a particular criteria, you can, for example, deselect that option, and you can also enter in a particular note. And so if a colleague were to see this similar variant or a similar gene, they'd be presented with your interpretation as well as your notes for downstream analyses. And lastly, you can click on the save option, and again, that will store those for downstream interpretations. So beyond the ACMG classifier, we can see that this is a de novo variant. So this is showing us the associated samples in the project. I think it's kind of a unique feature, or a feature that I like, because we have nice graphics in terms of whether the variant is inherited from the father in a heterozygous state or a homozygous state, or similar for the mother. We can see in this example, it's a de novo mutation. We have the variant calling Q&R, which is pulling in relevant information from your VCF file. So if you want to go in more detail and look to see if there's maybe some strand bias, et cetera, or look at the variant allele frequency, you do have the capability to do so. But one of the highlighting features of the Geneyx analysis is definitely our phenotype-driven interpretation tool. So with the phenotype that we've entered, which is tuberous sclerosis, we can see that this particular gene has a very high score relative to the other variants that are present in this particular VCF file. So as mentioned, you can go in and click on these. And what it does is it's utilizing the available biomedical literature and identifying based and highlighting those key search terms. And so in this case, we can see with the tuberous sclerosis, we have information being pulled in for the TSC1. But if we scroll down, we can also see those relevant publications for the TSC2. So in this example, we have information being pulled in from OMIM, Uniprot, Decipher, and it's highlighting those key search terms, but it's also providing you with a hyperlink so you can go in and investigate these in a little more detail. And this would be, if maybe you're a clinical geneticist or a clinical expert and you want to kind of deep dive into the relevant information, really we're providing you with a consolidated view of all of the information available. But as mentioned, this is all being integrated into a phenotype output score. And so you can strictly use this. Alternatively, we're providing you with information being pulled in from ClinVar. So if I click on the pathogenic information, we can see that this particular variant in the TCS2, it's an intron variant, has been associated with a tuberous sclerosis 2 with a pathogenic and a 1-star review status. If you click on these dropdowns, it'll provide you with the hyperlinks. So for an example, if we click on the hyperlink, it will bring you to the relevant publication source, which again, is just really providing you with all relevant information in terms of making a complete diagnosis of the case. Beyond the ClinVar, we will also display information on the in-house allele frequencies. So in terms of making maybe an internal case diagnosis on the variant or gene level, we'll present you with that interpretation as well as the allele frequency. So in this case, we can see that we have an allele frequency for a given variant. It's observed in these 3 VCF files. If we want to go in and investigate those as well, you have that capability to do so. We also display the effect and prediction, which will pull in relevant functional and in silico prediction annotation sources. So we provide you with that relevant information. And then we also display the frequency. So the frequency, obviously, there's a lot of public annotations that allow you to determine the frequency of the variant in a given population. As a nice kind of visualization capability, if you click on the particular frequency, it'll give you the distribution of the observed variants in nomad exomes as well as nomad genomes. So in this case, we can see that we have 3 variants that are observed in the European non-Finnish and the observed allele frequency. So as mentioned, ultimately what we're doing here is we've quickly identified the candidate variant. One of the things in terms of an automated workflow is maybe implementing a particular gene panel. So just to kind of show you what this looks like, you have the ability to integrate a gene panel in your protocol. So even prior to uploading your variants, or you can do it within your particular project. Just to kind of show you how to do it within a project, you can click on the plus option. This will bring you to your gene panels. And so if we wanted to select our tuberous sclerosis here and click save, now we'll see an updated variant annotation where it's basically quickly identifying this candidate variant. And again, this gene panel is relatively small because it's only associated with 2 genes, but obviously you can see the utility if you have a larger gene panel. And so as mentioned, we do have really nice reporting capabilities. So if you wanted to report on a particular variant, all you have to do is go up into the right-hand corner and click on the report preview option. And this will bring you to the report editor. Again, what it's doing is pulling in all relevant patient and clinical information. If we scroll down, we can also see we have the particular variant of interest, as well as information being pulled in on the mother and the father, as well as the publication sources. And so, as mentioned, really these can be fully customizable. You can choose what information you want to have displayed in your report. Furthermore, when you click on the save option, it outputs into a PDF and Excel format, and then you can also integrate this with your LIMS or electronic health record system. So this was obviously kind of a straightforward case of this tuberous sclerosis. Again, this would be maybe a little more specific to target gene panels or whole exomes. But if I'm jumping back here into the presentation, we can now focus on the next case. So again, this was kind of a straightforward case diagnosis, but the next one is on Parkinson's. And so this was actually performed by a group that uses the Geneyx Analysis platform. And it was provided in a publication by Brains Communication in the last year, so 2021. And this was done by the Hadassah Hebrew University Medical Center. And so what they were doing is they were looking at a cohort of 12 individuals, or 12 siblings. 4 of those siblings had early onset dystonia Parkinson's. And so dystonia Parkinson's is a continuous or repetitive muscle twisting, spasm, or cramp. I provided kind of an image here on the upper right-hand corner, just to kind of show what those characteristics look like. Ultimately, what they did is they took these affected siblings and they performed exome and CNV sequencing. However, the results were inconclusive, so they weren't able to identify candidate variants that were shared among all of the siblings. So what they did is they ran a linkage analysis, and through linkage analysis, they identified a roughly 10 kilobase run of homozygosity in chromosome 6. And chromosome 6 is important because it actually contains a gene associated with Parkinson's, which is the PRKN. And just kind of some general information on PRKN, 60% of the PRKN associated Parkinson's are a result of copy number variants or structural variants. And so what they did is they wanted to further identify kind of the candidate variants, especially because they knew they had this run of homozygosity. And so what they did is they utilized one affected individual and a healthy individual. And just to kind of preface, the healthy individual had the same runs of homozygosity, but within the PRKN, they observed a meiotic recombination. So it wasn't completely similar to the affected individual. So what they did, because whole exome and CNV sequencing was inconclusive, was they decided to implement whole genome sequencing, and then they analyzed the output with the Geneyx Analysis platform. So in this particular workflow, we're going to be looking at a healthy and affected individual. So if I jump back here into the dashboard view, again, we have our Parkinson case, but one of the things that I want to kind of showcase was the audit trails that we provide. So if you click on the analysis, it'll bring you to your analysis details. This is kind of a great resource because it will show you the case in terms of who created it and on what date, the protocol that was implemented, the different genetic models, so we can see that we will be looking at CNVs. And then we also have some agnostic information. We have the phenotype that we've entered, as well as when the last modification to this analysis was made. On the right-hand side here, we can see we have our different samples that we've imported. So we have our proband, as well as the associated unaffected sibling. And then you can see here, we have our gene panel, which is integrated into the protocol for Parkinson disorder. And so what I'm going to do is click on the Analyze option. And again, this is going to bring up the variant dashboard. So the protocol in this particular workflow integrated a target gene panel. And ultimately what we're doing is we're trying to identify a variant that is not observed in the sibling. And so with this, the gene panel identified 4 candidate variants, which are pretty easy to kind of rule out. So the first one is in the SPR gene, and we can actually see that this is observed in the sibling, so we can easily eliminate that. The next one is in the CS F1R. Just by kind of a brief glance, we can see it's associated with a pretty low phenotypic score. But if we want to investigate this on the gene level, again, we can pull in that information from OMIM as well as Decipher. Both of these databases are indicating that this particular gene is associated with the brain abnormalities, which is also being supported by evidence being pulled in from ClinVar. So it's not necessarily associated with this early onset Parkinson disorder. Furthermore, we have the ATXN1. This is generally a variant that is observed in, or just repeatedly observed. One of the nice things about this is we can easily rule it out based on the allele frequency across our internal databases. So just kind of a nice utility of having your internal population database being displayed to you. And then the last one is in the SGCE, which is autosomal dominant, but we can also see that it's observed in the sibling. So we can easily kind of rule out these candidate variants. And so, as mentioned during the presentation, this group performed exome sequencing and CMV analysis, which provided inconclusive results. And so they performed the linkage analysis to identify that run of homozygosity, and they identified that chromosome 6 was of primary importance because it had that PRKN gene. So if I go into the CMV genetic model, what we are displaying is we have all of our CMVs, which are listed again on a per row basis. You can look at the location. So if you want to view this information in GenoBrowse, you have the capability to do so. Beyond this, we will display the length. So here we are looking at a 77 kilobase event. It doesn't overlap a repeat region. And we can also view the information to determine if it's being inherited by the sibling. So if I click on this for option, what this is doing is it's showing us that in the proband, this particular event is observed in a homozygous state, and we have a break end. So it's very similar to an inversion event. Similarly, we have the sibling, but the zygosity is heterozygous, and there's actually a full overlap. So this is already pointing to the likely candidate variant. Further supporting evidence, we can see it has a high phenotypic score, and it's also observed at a low frequency in public frequency databases. A nice utility about the CMV interface too is it breaks it down, breaks your events down based on the genes. And so in this case, we can see that we have our PRKN gene. It's associated with an autosomal recessive disorder. If we click on this example, we can see for this particular gene, it has been associated with the Parkinson's disease, especially in a juvenile onset in an autosomal recessive inheritance. We can also see that information being pulled in from the ClinVar. So we can see there's Parkinson's disease as well as the young onset. Beyond this, we have the phenotypic information. So if we click on the phenotype for this particular event, we can see that one, it's associated with Parkinson's disease. We have one according to UniProt for late onset. But if we scroll down, we also have information being pulled in for autosomal recessive juvenile. So again, this is providing you with consolidated relevant information. You can use these hyperlinks out to do kind of a further investigation. And then lastly, we have information being pulled in from ClinVar, which is indicating that we're looking at a pathogenic, likely pathogenic CMV in Parkinson's disease. So ultimately, we've quickly identified the candidate variant. In terms of the publication, this was actually the event that they observed. So it was a 77 kilobase inversion of that contained the PRKN gene. The publication also mentioned that there was a 50 kilobase event, which we can see here. However, this was ruled out because it overlapped a repeat region, and it was observed at a relatively high frequency in the public databases. And so in terms of the case diagnosis, we were able to quickly identify that this event was the candidate variant. And if we want to go in and generate that publication or that report, again, you can go into that report preview, and it will provide the report editor, which you can then make modifications, or you can, kind of customize this as you see fit. So this was just kind of an example of an easy way to use the Geneyx Analysis platform for both kind of a difficult case as well as more of a straightforward case. And so to kind of bring this full circle, I just wanted to focus on the conclusions from this study. So here, the Hadassah Hebrew University Medical Center, they were able to identify this runs of homozygosity, and through using the Geneyx Analysis platform and implementing whole genome sequencing, they were able to identify this 77 kilobase inversion. They further then utilized Sanger sequencing and identified the breakpoints, which happened to occur in exon 5 of this particular gene. And so the case conclusion from this is really that for undiagnosed young onset dopamine responsive dystonia or young onset Parkinson's disease, they suggested performing whole genome sequencing. But what we can take away from this is that the Geneyx, is really a great resource for those challenging undiagnosed cases. And as you saw, we provide you with a variety of consolidated and comprehensive information. So to kind of provide an overview of what I demonstrated today, really the Geneyx Analysis platform can improve your diagnostic yield for challenging undiagnosed cases. So again, maybe for the clinical geneticist or expert in the field, really we're not trying to replace your current analysis method, but maybe if you have some cases that are being relatively challenging with your own bioinformatic pipeline, we might recommend you trying our software. In terms of the workflow automation, this is a scalable solution for those more straightforward diagnoses. So it's a fast, scalable, robust solution. Prime examples could be autosomal recessive diseases, cancer, nutrition, gene panels. So there's a variety of different applications that can be performed with the Geneyx Analysis software. Just to reiterate, we have some key features which we demonstrated today. So we have up-to-date annotation sources with repeatable workflows, which allow you to obtain your consistent results. We have a powerful phenotype-driven interpretation algorithm, which allows you to quickly identify the candidate variants. We automate the ACMG classification with the ability to perform modifications and customizations. And then we also offer simple reporting and integration with LIMS and electronic health record systems. And so if you're interested in the Geneyx Analysis platform, I would recommend reaching out to Kim, myself, or support at geneyx.com for a free consultation. And so with that brings us to the final slide of this presentation. So I wanted to just say thanks for attending. Again, as Kim mentioned, we will provide you with a recording. So it's going to be made at the end of this webinar. We'll send it to all of our attendees and likely put it up on our website. And then we do offer the free demo trial. So if you wanted to see kind of the applications of the Geneyx Analysis platform, you could easily send us your 3 of your most challenging cases. We'll solve them and review the data with you. Or you can open up a trial account and process your cases yourself with no charge. But again, if you have additional questions, please don't hesitate to reach out to Kim, myself, or support at geneyx.com. And so with that, I think we'll kind of transition into the Q&A. I know we only have a couple minutes, but I think Kim has probably flagged a couple questions.
Hi, Eli. Yes, thank you, Eli. Great job. There have been a few questions that have come in. The first one for you is, do you have workflows for cancer?
Yes, so that's a great question. We actually have protocols that are specific to somatic workflows. So for example, if you wanted to work with a tumor normal or cancer only, we do have protocols that can assist in that. And we provide you with relevant cancer databases, as well as if you want to integrate your own customized database, you have the ability to do so.
Okay, second one that looks like came in, can data be shared across different labs that use Geneyx?
Yes, so it really kind of depends on how your license is set up, but the Geneyx Analysis platform does support inter-lab sharing. So basically, if you provide access to other labs, they'll have access to your frequencies, your variants, your annotation sources. So the answer is yes, we would just have to kind of set that up on your license.
Okay, I mean, it looks like we have time for one more. Does Geneyx Analysis have an API interface?
Yes, so if you wanted to, you could, we can definitely reach out and talk to you directly, but we actually have some script repositories on GitHub, which allows you to basically automate the processing of FASTQ, integrating it into the Geneyx Analysis platform, as well as integrating with LIMS or electronic health record systems. So we can talk directly, but the answer is yes, we do have an API integration.
Great. We're at time now, so a couple of other questions have come in, but I can definitely pass those over to Eli. We can answer those and then send those out to you guys after, along with the recording. Again, I say the same. Thank you, everyone, for attending. We would truly appreciate it. And please reach out to either Eli, Kim, support. We're happy to help. And look forward to more of these to come.
Perfect. Thanks, Kim. Thanks, everybody.
Thank you.